Okay, wow, that was a, I just want to say thanks so much, Alejandro and Luis, and to the whole IUBMB for this incredible honor. And it's especially nice to receive this in such a warm community. I think this meeting over the last few days has been just fantastic, and it's in obviously a gorgeous place that's spectacular, and it's been especially nice just because of the amazing community that it's brought together. And I've just had an amazing time, and I'm just really thrilled and happy to have shared these last few days with everybody. So what I thought I would talk about tonight is sort of some new work that we've been doing, or really a new direction for the lab. It takes the ideas of synthetic biology, but tries to start to apply them to therapeutic problems. And so that's why I have my little, oh, you're not even looking at that. Okay, let's try that. Okay, okay. So it's the idea of trying to make biological circuits that have therapeutic functions. And I'm going to try to define this really precisely as we go. And this is really exciting for me. It's about probably the second time I've really talked about this work. So I'm excited to share it with you. So the starting point for this, and really I think for almost anything we do in my lab, is this view of biology as from the circuit level. Okay, most of the functions that we're really interested in organisms they really arise not because there's one gene that does it, it's because there's a circuit of many interacting genes and proteins that together implement that function. We've seen that throughout this meeting with stress response circuits that we've heard about, and circadian clocks, and vernalization, and all sorts of different systems. And our challenge is to really, in biology, is to really understand how these functions arise from this web of different interactions. But whether it's interactions between cells, like in the immune system on the right, or interactions between molecules within a cell on the left, at least for me, I can never stare at these diagrams long enough and hard enough to just see how they work and really understand why they're wired up that way instead of some other way. And so, as Alejandro mentioned, many years ago now, let's not say exactly how many, I tried to do something else. As a grad student, I was finishing my PhD with Stan Leibler, and I thought, well, maybe instead of trying to, take apart a natural circuit, is it okay? Oh, I have a pointer. Yes. My lightsaber. Okay. So instead of doing that, maybe I could just try to make some simple function. And I was reading a lot about circadian clocks that we've been hearing about at this meeting. And I thought, couldn't I just try to make the simplest thing I could imagine, but program it myself? And then I would understand, I'd know all the interactions because I put them in there myself. And I'd know if they were really sufficient to get it to do something. And so this was just trying to do that. So we took 3 repressor genes, hooked them up in a game of rock, scissors, paper, each one repressing the next one in a little negative feedback loop, put them into E. coli, and indeed they generated these oscillations, as you can see in the movie, in individual E. coli cells as they were growing. This was a very exciting moment for me, just to say that you could just make something simple and it would actually work in a cell, something I did not take for granted. But you can also see immediately that there's something interesting that was not designed in the circuit. And the interesting thing that was not designed is the variability. So 2 sister cells or cousins, they start to go out of phase with each other with this oscillator, unlike a real circadian clock. That happens relatively rapidly. And that immediately provoked now a more general biology question. Well, where does that variability originate? Not just for a synthetic circuit, but for any circuit. And so we started to think about how could we, maybe it's just the stochasticity, just random fluctuations that are involved in gene expression processes that could generate some of this variability. And we came up with a way to try to measure that by putting 2 different fluorescent proteins under the identical control so that if there were differences in the expression between them, more red or more green, you could use that to measure the amount of noise or stochastic fluctuations in gene expression, how important that was. And that led then someone else, Johan Paulsen, to go back to the repressolator and understanding more about noise, try to really understand where the variability came from. And so this is from Johan's 2016 paper, where he took the original repressolator, but he realized something, which was that one of the interactions, one of those repression steps from TET-R to lambda repressor was too tight. The DNA binding was very tight, and that made it actually very sensitive to stochasticity. And by just effectively weakening that interaction and doing a few other little tweaks, he made this repressolator into something that works incredibly precisely. This is a little chymograph on the bottom here, and you can see over time how periodic it becomes. On the top, you can see it going from red to blue to green and so forth over and over again. Sorry, I should say that that movie is one cell trapped at the end of a channel, so all the progeny kind of flow out to the bottom. And so if you look at the top, you're looking at one cell over a long period of time. And so he discovered a kind of design principle of how to circumvent noise, and got it so good that it only loses about a half a period of precision over 180 cell cycles. And what this tells us is that to make something that operates precisely in a cell, you don't need a huge complicated circuit to get precision. Simple circuits can be amazingly precise. And so this is just an example of what I think is a virtuous cycle in synthetic biology. We try to build something just to see if it can work. It provokes new biological questions. We try to address them, and we come back and we can actually re-engineer a system and make it work better. And we learn something about biology in the process. Now, since then, throughout the synthetic biology community, we and many others have made all sorts of different functions. This is just a very small subset of them. And in all of these cases, I think, there's something that we learn, and I put here little design principles that have emerged from all of these different examples. So we learn something every time we do this. And so I think what's happening in synthetic biology is that we're kind of evolving over time from a phase of just trying to see what's possible, what can we make happen in a cell at all, to a phase of discovery, what are design principles that these synthetic circuits reveal, and then possibly to a third phase, and of course these are all going on concurrently, but in principle, a different phase where we actually try to turn it into engineering and solve real problems. And okay, so what problems can we solve now that we know that we can make circuits that operate very precisely? Well, if we think about medicine, drugs are one of the dominant paradigms in medicine, and they're very effective at killing cells, at inhibiting target proteins, or replacing enzymes or hormones. Very effective. But a circuit can do things that no drug could do. A circuit, and I'm showing here a cartoon of the p53 circuit, which is the way our bodies naturally suppress tumors, not with one tumor suppressor protein, but with a circuit of tumor suppressor proteins. These can kind of sense the state of the cell. They can take in lots of different kinds of information, integrate it, make decisions, and control all kinds of cellular functions. They're really powerful that way. And so the question we've been wondering about for a long time is, could we start to make therapeutic circuits that are synthetic but provide some of those capabilities? And I want to show you two examples today that are steps in that direction. And the first one really focuses on the question of specificity. How can we specifically target, in this case, cancer cells? And the second one is on the question of precision. How can we make circuits that operate in vivo very, very precisely? And that'll be for gene therapy applications. So let me start with the first of these two projects. So, as many of you know, cancers are driven by oncogenic signaling, and one of the classic oncogenes is RAS, mutant RAS, and that drives survival signaling that causes proliferation through many different downstream signaling pathways, as you see here. Over the last decade or so, people have developed incredible inhibitors that can inhibit RAS activity, things like sotorasib that inhibits G12C, or RMC-7977 is a pan-RAS inhibitor that can inhibit all RAS signaling. And these block RAS signaling and they suppress the cancer, which is great. However, they're susceptible to resistance because you can get mutations either in RAS itself or in downstream pathways that circumvent the drug. And so this is kind of a fundamental limitation of this inhibit approach. And so what we wondered is whether there's a different approach, which is instead of trying to inhibit the oncogene, we simply try to put in a circuit, and I'm going to tell you what the circuit is, put in a circuit that can sense that the cell has the oncogene and is expressing it, and then immediately and directly kills the cell. A very simple kind of circuit, sense, kill. So here's how we imagine this working ideally. We imagine we would encode this circuit as mRNA and deliver it to the tumor and adjacent normal cells in lipid nanoparticles, like a COVID vaccine. Then it would go in, it would sense, the protein circuit would sense that it's in a mutant cancer cell, kill it, and then the mRNA and the protein would all go away, and your normal cells would be left intact. This would be great. Okay. Now, that's very nice, but in reality, there's lots of challenges to get there. So, for example, we need this circuit to be very sensitive. It has to activate in every cancer cell. We need it to be very specific. It should never activate in a normal cell. And we have this problem of delivery, which is a classic problem across many areas of biomedicine, which is how do we get this circuit into most or all of the cancer cells anyway? Now, a few years ago, we had started thinking about these things with a very contrived and very synthetic, very artificial system of engineered proteases, which we were using to see if we could make a little toolbox that we can make protein circuits that would do anything we wanted. And it was, we had a little demonstration with a RAS killing circuit, but it was not at all realistic. It was really just a proof of concept. But then I've got a kind of new generation of people in the lab. Andrew, an MD PhD student between UCLA and Caltech, Lucas, an incredible grad student, and Xiu, who's a postdoc who had previously studied pyroptosis that I'll tell you more about in a second. And they started really trying to dive in and say, how do we make this thing into something that really could work for real? Okay, so first of all, let me tell you now what this circuit is. So if I have RAS, wild-type RAS is generally unclustered on the membrane, but mutant or active RAS clusters, and that gives us a way to sense RAS activity. If we take a protease, split it into two halves, fuse each half to a RAS binding domain, then in the active or wild-type RAS, or mutant RAS condition, that's what cluster the two halves of the split protease together, reconstitute their activity, and if you have also a proteolytically activatable cell death effector, then you can have reconstitution of protease, you can have, detection of the mutant RAS, cause reconstituted protease, and have that cause activation of cell death. So it's a very simple circuit. Okay, and we built this, and it worked great when we put it into HEC293 cells that were overexpressing the mutant RAS target. It did not work at all when we put it into actual cancer cell lines that expressed endogenous levels of mutant RAS. It was not sensitive enough, and we knew more engineering was needed. And so we went on a little campaign to engineer every aspect of this system. So first we did a screen of all kinds of different RAS binders of various types, and we expressed them fused to the two halves of the split protease along with mutant RAS and a reporter. And we use this technique, if anybody, I don't know if anybody else in the audience uses this polytransfection. Anybody? Nobody? It's the most useful and underappreciated approach. It's simply if you're going to co-transfect things and you don't want to do it in a way that's correlated in expression, you want to explore a broader expression space, you can first encapsulate in lipofectamine and then mix rather than mixing the DNA and then encapsulation. A very simple idea due to Ron Weiss and something that's very effective for quickly exploring a big expression space. So we did this scan. We did it of natural protein domains, de novo designed binders and antibodies and other things. And I should say in this plot, what you ideally would like to have is a good sensor would be highly responsive on the y-axis to mutant RAS, but not responsive to wild-type RAS on the x-axis, so the top left corner there. And we did find binders that fit the bill, and the one that really worked the best is a monobody called 12VC1, and it was designed exactly for this property to be very specific to mutant RAS and to bind it very specifically. It also binds to other RAS mutants as well, but not to wild-type RAS. And so that's the sensor. That's improved the sensor a lot. I'll show you that in a second. We also wanted other ways to improve sensitivity. And the beautiful thing about engineering proteases is that you can start to make little amplifiers. And that's not totally contrived, because if you look in a lot of natural proteolytic circuits, like cell death pathways, they have protease amplifiers. It's a natural system. So we tried to engineer those. We made a lot of different designs. And that's why this slide is known as the gallery of suffering, which I insist I must have a gallery of suffering in every talk, because Andrew went through a lot of different designs, and I can't tell you that at the beginning we had any idea which of these, if any, would work. And in fact, most of them failed completely or even gave us weird behaviors where they anti-amplified, they de-amplified. But one of them did work well, and that's the one that I'm showing you here, where you take two halves of the split protease, each one is caged by a mutated version of its own half. They're attached to coiled leucine zippers. Oh, I guess I have to do it this way. These are these light blue domains here. And as a result, if you cleave with an input protease, then that releases the two halves, and they can be brought together by these complementary leucine zippers, and then that activates them. And this actually worked really well, so it really amplifies the input, and it doesn't give us a high background. So that was great. The last little engineering trick we did is we stole an idea from viruses, and to have really good stoichiometry between the two halves of our sensor, we encode them as a single polypeptide, and because they have a protease that's encoded within them, that protease cuts itself out of the nascent polyprotein. So we just cheated and plagiarized all the viruses in the world. Okay. And all of those things together actually helped a lot. So how well do these circuits work? So let me show you a very simple version of the circuit. This one is actually without the amplifier. We take the sensor and the effector, these two proteins, encode them in mRNA, encapsulate them in lipid nanoparticles, and then just put them on cells that are either mutant RAS or wild-type RAS. These are 3 cell lines that are wild-type RAS. And what you can see here on the y-axis is that they're not getting killed, even as we increase the concentration of the mRNA LNP that we were using to deliver them. Now, if we do the same thing in RAS mutant cell lines, these are G12C, for those who know RAS, you see that we really efficiently and completely kill them. That's nice. Now, one thing people always ask is they say, well, wait a second. If you're putting this thing in and it's detecting RAS activity, there's natural RAS activity, so maybe you're going to kill those cells. So here's one reason we think that this sensor is specific enough that that's not happening. If we add EGF in cells to induce RAS signaling, we can actually induce, ERK activity here with the blue curve, but we don't induce cell death. So we think that it's specific, and at least in the cases we've looked, we don't have to worry about that. Okay, now the immediate question is, how does this compare to drugs? I told you before, there's sotorasib, there's these pan-RAS inhibitors. They're really revolutionary and sold by Revolution Medicines in some cases. And so we wanted to do a kind of King Kong versus Godzilla experiment of drugs versus circuit head-to-head, okay? And so here's what that looks like. So here I have these 2 drugs in the blue and the purple. And what you can see is that if we put them on HEC293 cells, which are wild type for RAS, as you would expect, they don't do anything. The cells are viable. If we then go to other cell lines that are RAS mutant and are addicted to RAS, meaning they depend on the mutant RAS for survival, then it kills them pretty effectively, as it should. Okay. On the other hand, if you take other cell lines that are also mutant RAS, but are not RAS addicted, these drugs have more modest effects on them, okay? And that's because the whole mechanism of the drug is it inhibits the pathway, and that only does something if downstream the cell really requires that signaling. If it doesn't require it, it's not going to kill the cell. Okay, so let's see what the circuit does. So with the circuit, we retain specificity. We also don't kill these RAS wild-type cells, but you can see that we effectively kill both the RAS-addicted and the non-addicted lines. And that totally makes sense. Again, that's just because all the circuit does is it just asks if RAS is mutant or not and kills it if it is. It doesn't care about addiction or any of the downstream circuitry. I think a nice and really vivid way to see this is to co-culture RAS wild-type and mutant cells. So here the wild-type cells are green, the mutant cells are in magenta. And you can see with a vehicle control, they both grow. With a chemotherapeutic, it kills everything. The drugs preferentially kill the RAS mutant cells, as you would expect, but the circuit is more specific and more potent in its killing than the drugs. Now, the thing that's kind of really nice about these is we start to think about resistance, because I told you at the beginning, resistance is really a big issue with these drugs. And so to try to kind of think about that, we started culturing, these are MIA packet 2 cells, these are RAS mutant, and we culture them for 3 weeks on sotorasib. And when we do this, as you would expect, we select for resistance. And you can see that because the dose response curve here is shifted out about 30-fold from where it would be. And in fact, they're so resistant that we can't really kill them all 100%. But now, if we take those resistant cells and we treat them with the circuit, we find that we have, the technical term is negative cross-resistance, meaning that instead of being less sensitive to the circuit, now they're more sensitive to the circuit. One explanation for that could be that a common way to get resistance to the drugs is to get amplifications in the RAS gene, and those would naturally make it easier, not harder, for our circuit to detect RAS. Okay, and when we do the reverse experiment and we try to culture on the circuit for a long period of time, we don't see the same kind of effect. So we see only a tiny, very modest 1.3-fold effect in terms of resistance. And we don't see any change in drug sensitivity. Okay, now what I showed you so far is we can get really high with this very simple circuit, but a lot of engineering. We can get very high specificity and sensitivity, and that's good. But everybody in the back of their mind may be thinking, big deal, you'll never get those into the cells. Okay, and that's a reasonable concern. So how do we deal with this? And this is where this is a very, science is very serendipitous at times. And a few years ago, got an application for a postdoc from Shiyu Shah. And he's somebody I wouldn't think would normally be interested in my lab, structural biologist. And in his PhD at Harvard Medical School, he had solved the structure of Gas-Dermin D, which is the effector protein that initiates a cell death program called pyroptosis. Pyroptosis is interesting because in contrast to apoptosis, which is immunologically cold. It doesn't stimulate the immune system. Pyroptosis is immunogenic. It releases IL-1 and a bunch of other cytokines, as well as damage signals that generally recruit the immune system. And what other people have shown really beautifully by a few different groups now is that if you activate pyroptosis in a minority of cells, like even just 10% or 20% of cells in a tumor, in some cases that can recruit the immune system and clear the entire tumor. So this was like perfectly, exactly the thing that we felt we needed. So could we make the output pyroptosis rather than apoptosis and then get a non-cell autonomous spreading of the effect of the circuit? And the other fortuitous thing is that these gasdermins that are the effectors of pyroptosis are also naturally activated proteolytically by cleavage. And so the obvious thing is to replace their normal cleavage sites with cleavage sites for TEV, which I should have told you before is the protease we've been using for a lot of this. Okay, so those effectors, so Shiyu engineered them and they work really well. What you can see here is that black means we're killing the cells and we're doing it when they're in a mutant RAS cell line and we have both the effector, the cell death effector that's engineered and the sensor, not when you were missing either one or the other, and they're not killing in the wild type RAS cell line. There's other advantages outside of the synthetic paradigm for these circuits because they can selectively activate pyroptosis just in cancer cells to avoid hyperinflammation. And they also can overcome a problem that you have in real cancers, which tend to sometimes silence the endogenous gasdermins because they're trying to avoid pyroptosis. Here we're supplying gasdermin ourselves as part of the circuit. So there's some nice features of that. Okay, the last part though is really, can these things actually work in vivo? And so for this, my lab's not really an in vivo lab, although we occasionally dabble, usually to my detriment, but it has happened on occasion. So we collaborated with an amazing scientist, Hao Zhu, who's an expert on liver development, liver cancer at UT Southwestern, and with Dan Siegwart, who's an expert on, also at UT Southwestern, expert on lipid nanoparticles, and his student, Stephen Moore. And we wanted to do an experiment. We did a variety of experiments in mice, but the one that I'll show you right now is one where we induce tumors with hydrodynamic tail vein injection of a activated RAS construct and a p53 hairpin to knock down p53. This is like a multifocal liver tumor model, and it generates a lot of tumors. And then we systemically treat with a lipid nanoparticle mRNA containing the circuit on days 5, 6, and 7, and we look day 26 at the livers to see what happened. Okay, so these are the untreated. The model itself has some variability, and so if we rank them from the most gross, the most yucky livers here on the left to the least on the right, you can see there's some variability. Those are untreated, but the treated ones, you can see they're not 100% perfect, but they really suppress effectively tumor development, even when you're delivering the circuit, after the initiation of tumors. Now, there's a lot of questions we want to know, like how much can we delay the treatment and so forth, so we're working on those things too. I should also show you, if we just quantify the tumor area, you can see this is untreated and this is with the circuit, so tumor area is really suppressed across the different mice. And if we look at liver weight to body weight ratio, which is a metric for liver health, that's restored to a healthy value. Okay, so that's the first part of the talk, and just let me conclude that part. So what I told you so far is that this is a very simple synthetic circuit. It's not pushing the edge of complexity. But you can take a circuit, you can encode it in mRNA, deliver it in lipid nanoparticles in vivo, and it's kind of a different kind of therapeutic. It's fundamentally different from drugs because sensing and killing is not the same as inhibiting, and there's consequences for that with respect to resistance and addiction, independent of RAS addiction. It can target drug-resistant cells as well as drug-sensitive cells, and we think it's less susceptible to resistance. And I think the thing that makes me really excited about this work, which I'll come back to at the end, is that there's nothing here that's very RAS specific. So we have a sensor for RAS, but we could swap that out with a sensor for any other oncogene we want. We don't have to detect one oncogene. We could detect markers of cell state. We could detect multiple signals. It's really modular and programmable. And so, we're trying to go deep right now on one disease, but I think in the end we would like to also be able to generalize it. Okay, now I'm going to switch gears to the second story. So that was about specificity, maybe, and this one is going to be about precision. Okay, and I want to start with reminding you, I don't know if many of you have heard of a disease called Rett syndrome. Rett syndrome is a really severe neurodevelopmental disease. It was discovered by Andreas Rett in Vienna in the 60s, and in the US it occurs in about 1 in 10,000 female births. It's X-linked. It's a disease in which the children develop normally for 6 months or even a year, and then they start to regress and lose abilities that they already had gained in terms of motor abilities, speech, things like that. So it's really very devastating condition. Huda Zogby in the 90s mapped this disease to mutations, de novo mutations in this gene MECP2. And then in the 2000s, Adrian Bird's lab showed that if you restored wild-type MECP2 expression after the onset of symptoms, they could be reversed. And that made this a great candidate for gene therapy, except for one thing. Okay, except Huda and others also showed that it's extremely dosage sensitive. So the reason it's in females and not in males is because females are mosaic for the X, and if you have a loss of function mutation in one copy, that gives you the disease but doesn't kill you. Males who have one copy of the X lose it completely and die before birth. So loss of one copy is deleterious, gives you the disease, but an extra copy gives you MECP2 duplication syndrome, which is another disease, has some overlapping phenotypes, and is also pretty bad. And what that means is that you really, if you're going to do gene replacement therapy, you want to tune the expression level of the ectopic MECP2 into a really narrow therapeutic window. And this is extreme in Rett, but it's present in a lot of monogenic diseases where you really need to not overexpress or underexpress the therapeutic gene. Now, the reason that's hard is because the way we deliver genes right now is with these AAV vectors, which have been going through a little revolution in terms of their engineering. But we can't, we can control the dose, but we can't control exactly how many copies of the gene get delivered to any particular cell. You might have some regions of a tissue that get more, too much, too little, and there's just stochasticity or random uptake. Different organs might get too much or too little, and so on. And so I was telling somebody just here at the meeting that you know, at our Caltech biology retreat, my colleague Mitch Gutman had just come back from a Rett syndrome meeting and said, you guys should do some kind of circuit because they have this overexpression problem. And so I said, okay. So and Mike Flynn, who's on the left here, was at that retreat, and he was like, yeah, let's try and do that. And then Rong Du, who's an amazing student, joined the project a little later. Okay, so how would a circuit help with this problem of controlling expression? Well, think about it this way. What you'd really like, ideally, is something where you could crank up the dosage of the gene therapy, but not the expression level in any particular cell. So you'd really like a curve like this green curve, where you can just turn up the dosage, but make sure you go to kind of a flat level here, independent of dosage. Okay. Obviously, if you put nothing in, you're not going to get anything. But as long as you get it high enough, it should become dosage independent. That's what you'd like. And there's lots of other features you'd like as well. Ideally, you'd like to be able to tune what that level is to match it to the disease, and so on. Okay, and there's a classic circuit motif in systems biology discovered by Uri Alan called the incoherent feed-forward loop. It comes up in all of these different circuits all the time. These incoherent feed-forward loops are systems where you have some upstream factor that activates something that activates the response you're interested in, but also activates a negative regulator of the same thing. So it's incoherent through two paths. And this kind of circuit can, in general, under some conditions, give you this behavior. Okay. But not always. It depends on the relationship between that negative regulator and the target. So here the target is our mRNA. And if this arrow here is too sensitive, if it's ultra-sensitive, or if it's not sensitive enough, that's not good. What you need is for it to be kind of linearly sensitive. So a linear increase in the amount of the regulator gives you a linear decrease in the amount of the effector. And if you're in that regime, then you're on that pink line, and the pink line gives you dosage invariance. But if it's too sensitive or not sensitive enough, you lose it. So just to say that it's not automatic that you get dosage invariance. Okay, so we started thinking about different designs to do this. At first, we did a protein level design, but then we switched to microRNAs because microRNAs are compact and can be embedded into a gene therapy and have other advantages. And so we actually made now a zillion different versions of these circuits. I'm going to just show you schematics that are one version, but there's many. In these circuits, you are co-expressing a gene along with an intronic microRNA, and the microRNA targets the gene at some target binding sites. Now, we first did this, by the way, I should say multiple groups have actually built circuits like this for us and explored a little, but they never really gained traction, and they weren't really that usable, and they hadn't been explored very quantitatively. So the first version that we built, we thought we were trying to get that linear behavior, so we put a single target binding site. We made it as strong as possible, 21 base pair, fully complementary site, and it did give us regulation. As we increase the dosage, we see we get reduced expression of the cargo protein. But this is not flat here. Okay? And so something was wrong. Luckily, the circuit has many adjustable parameters. You can change the number of binding sites for the microRNA, the degree of complementarity in the binding sites, and many other parameters. And the interesting thing that happened was, as we were going down in the length of the complementarity from 21 to 20 to 19 to 18 to 17, we get to 17, and with a single binding site, we lose regulation altogether. No regulation. But then if you take that 17 base pair site, which doesn't regulate, and you multimerize it to 2 copies, 3 copies, 4 copies, then regulation comes back. And when it comes back with dosage compensation. And that's what you see here in this flat region here. I'll just show you a better version of it here. This is now over 2 orders of magnitude of dosage variation, where there's just a few fold variation in the actual expression level of the protein. And so you can see this, we get dosage, we solve the problem. We have dosage invariance in these circuits. And of course, we had to make an acronym that's required, but we're fastidious with acronyms. They have to make sense, and no internal capital letters, you guys. Okay, so we call this DIMMER's dosage invariant microRNA mediated expression regulator, which actually makes sense if you think about it. Okay, so we solved the problem from an engineering sense. We could have been happy, but we were also kind of puzzled and disturbed because we don't really understand why this works. And in fact, it's the opposite of what I would have expected. I would have thought if I put multiple binding sites for multi-specific regulation, I would have made the whole thing more cooperative, and I would have gotten the ultra-sensitive regulation, which would have not given me this behavior, right? This is not what I wanted. I wanted it. It's good. It works, but it's not how I would have thought it would go. So we've been puzzled, and we are working our way through some biochemical experiments to really try and understand the principles. So the first thing, TNRC6 came up already earlier today in Gunther's talk, and what was mentioned is that TNRC6 is this disordered protein. I drew it incorrectly. I'm realizing from earlier today I should have drawn it more disordered, but it's a scaffold that can bind to multiple Argonaute proteins. And there's an inhibitor for it. There's a truncated version called T6B that's a dominant negative that inhibits binding to the real TNRC6 by competing with it. And we found that when we take these, the ones that work, these 4 copies of the 17 base pair binding site, and we add this dominant negative for TNRC6, we lose regulation. So we go from this nice dosage compensated behavior to something with no regulation at all. That tells us TNRC6 is required. Now, TNRC6 was not required when we had the single fully complementary 21 base pair site. It's something that's required in this regime where you have the weak multimeric binding sites, but not the single fully complementary binding site. So we know it's involved. We did a lot of experiments, too many to kind of summarize here. And we also made a model. And the model tries to think about, sort of a statistical mechanics model where we basically say there's a bunch of binding sites, and they can be occupied or unoccupied, and we keep track of all the different configurations, and we write down a big partition function for the physics people here, and then we can kind of play around with the model. And the models generally can explain some of the behavior, but I'm not going to go through it. So here's how we're now thinking about this conundrum. Okay, let's go back to the single site. So if I have a single fully complementary binding site, I have tight binding of Argonaute here with the microRNA, and that gives me efficient slicing. So I get very efficient destruction of the target mRNA. And if you do that in the model, you get a curve like this. So it's so efficient, the slicing, that it engages at a very low concentration of the construct, and it gives you dosage invariance, but it does it at a really low level. And then there's the tail. The tail inevitably occurs because your microRNA is binding to the target, but as you go up to really high levels of the target, you start to accumulate complexes that are bound but not yet degraded or not yet sliced, or they're sliced and not yet eliminated. And there's no way to fully get rid of that. Okay? So that's the behavior. And this actually, in other words, is working like we would have expected. But I want to point out that it's doing this in a low regime. And if this is so low that you can't even see the GFP that you're looking at, you might think you might not even see it. Okay. But it has that behavior. Okay, now what happens when we have multiple partially complementary binding sites? Now we come in, we have TNRC6 as scaffolding. The slicing is weak because the individual affinities of Argonaut for these individual binding sites are weak and they don't efficiently slice. But TNRC6 and the complex of other factors that it recruits compensate for that with deadenylation and other mechanisms to degrade the mRNA. So if you do this, then in the model, you get essentially the same shape curve. It's just that you engage at a higher concentration because the affinities here are lower, and, but otherwise it's the same. And as a result, you can actually get this dosage compensation behavior, but get it in the regime that's more appropriate to where you're targeting. Okay, that was a very hard to explain, long-winded thing. I hope somebody got it. Okay, we'll see. Okay, so we're still working on that, but I think we're really trying to understand this biochemically, and I think this is a great example of where the synthetic approach poses a question that maybe is similar to, but maybe it just comes at it from a different angle than you would if you were just trying to understand a particular natural system. Okay, so what's also great about this system, just practically, is that it's tunable. So by tuning the complementarity, 17, 18, 19, we can get dosage compensation at different levels. We also built a version of the construct that's DOX-inducible and gives us dosage invariant expression at we had a level that we can specify with doxycycline. It's portable. We get dosage invariance across multiple cell lines. Here's 4 different U2OS, CHO-K1, HEC293, N2A, and so on. So we see it working broadly because all it depends on is just the microRNA machinery. And it's also useful when you're making stables as well as transient transfection, because in stables, you usually get these are just here like 5 different clones where we stably integrated a fluorescent reporter. And you can see they're there, they're expressing at different levels. They're nice and unimodal, but when we incorporate the dimmer, the output expression from the dimmer is really uniform, irrespective of whatever random integration site you had in the stables. And finally, you can make many of these constructs with different microRNAs, because the microRNA coding space is very large. And so we played around with a little algorithm that gave us, in this case, 10 different sequences, but I think this is really scratching the surface. There's no real fundamental limit on how many orthogonal versions we can make, but this allows us to at least regulate 10 things independently if we want to. Okay, there's other things that I won't show you. We showed that by adding these circuits to constructs for imaging, CRISPR imaging, we can reduce the background substantially. We get much better imaging. We can do it with a base editor, and then we reduce off-target base editing. And we can deliver it in AAVs to mouse brains, and it functions in mouse brain, which is going to be important now. Okay, so having designed these circuits, can we actually do something with Rett syndrome or other gene therapies? And this is where, again, really fortuitously, Viviana Gradinaru is an expert on gene therapy, AAV capsid development, who happens to be on my floor at Caltech, kind of randomly. And her student Acacia is really fantastic. She was an MD PhD with USC and Caltech. And we showed them these circuits, and we decided to try to do the mouse model with them. And they had just come up with these AAV capsids that target the CNS and de-target the liver. So AAV9 is a particular capsid, but really accumulates a lot in the liver. Their B22 capsid goes to the brain and not the liver. And so we took these circuits, actually not the ones I've just showed you. We took an earlier version of the circuits, because this now goes, there's a new timeline here where I'm diverging because these experiments started before we fully optimized the circuits that I just showed you. So these are actually the 1.0 versions I'm going to show you now in the disease context, and the 2.0s are better. But anyway, we encoded these circuits, we put them into AAVs, and then we want to know, like, how do they behave in the brain? And the nice thing is we could just label by single molecule FISH the ectopic delivered mRNA for MECP2 and also the endogenous MECP2. We could look at them in the same cell together. And so when we do that, if we do an unregulated construct in these plots, the x-axis is the endogenous MECP2, the y-axis is the ectopic. And you can see with an unregulated construct without the circuit, you have a little more mRNA for ectopic than endogenous. But when we add a weaker version of the circuit or the strong version of the circuit, we can really suppress expression in the brain. Then once we could do that, then we said, okay, let's try to really do this in a mouse model of Rett syndrome. So we got these Rett model mice, and then Acacia did a behavioral experiment that lasted half a year, where she basically delivered the AAVs, in this case at week 0, and then did behavioral assays periodically going out to 26 weeks to follow these Rett behavioral phenotypes, things like hind limb clasping and other phenotypes that other people had previously identified for Rett. Okay, and here's the results. So the red group is the untreated. So the y-axis here is a Rett phenotype score. Higher values are worse, meaning more severe, and low values are more like wild type. And you can see the wild type mouse here has very low mice. These are groups of mice. The wild type mice have very low values of the score. The untreated, the score goes up over time in this red curve. Oh, sorry, I keep doing this with my mouse and you guys don't see it. Yeah, the red curve goes up over time. The unregulated, maybe, I don't know, has some benefit in the early period, but basically becomes sort of undistinguishable from the untreated. The weak circuit's sort of in the middle, but I think the strong version of the circuit that had the most suppression of MECP2 is giving us the lowest values of these phenotypes. And here I'm just showing you expanding out each of these groups into the individual mice within the group. And you can see the effects of the strong circuit are kind of felt across more or less all of the mice. There is kind of an uptick here at the end, but that's where we are. So we think we see kind of quite strong benefit in this mouse model of Rett syndrome, at least out to 6 months. Okay, so just to conclude this part of the talk, what I told you is that, I think, at least for me, when I started working in mammalian cells, I thought everything was just messy. And what I'm learning is it's not messy, we're messy. And when we do a transient transfection, that's messy, but things can be precise. And this is one way to make, I think, really precise controls. Something that's useful in research. I think whenever you want the convenience of transient transfection, but don't want to have a big mishmash of expression levels, want to have a precise expression level, you can just use this to get that, and you can even tune it to different levels. So I think this is a very useful little device. It improves the outcomes in this mouse model of Rett gene therapy, and so we're interested in trying to explore how it can be used in Rett and other gene therapy contexts. Okay, so I think I'm doing great on time. I have 5 minutes, is that right? Okay, so now comes I thought I would do a deep thoughts last conclusion here. So this has been, I wanted to do this talk where I start with the really old stuff with a repressilator at the beginning, and it's so primitive. And if you look at the circuits that I just showed you, the size of the circuit didn't expand very much. I had 3 genes 25 years ago. These circuits have 4 proteins. They're not getting much bigger, but they are getting much better. And we have much greater predictability, much greater modularity, and much greater, I think, understanding of what we're doing. And so I think the dream of synthetic biology has always been that we're going to create a kind of technology that's programmable, rational, it's going to operate in cells predictably, and that if we could do that, then things could scale fast, because we know technologies do scale. And I apologize for this, but I have to show you I don't know how many of you have seen this before, what the first transistors looked like in 1947. Who's seen these pictures before? Anybody? No? Yeah, look at this thing. Okay, this is the first transistor, 1947. Okay, Bardeen got 2 Nobel Prizes, one of them for that. And it looks like a mess, okay, much like some of our early synthetic biology circuits, I would say. And then in the 50s, they start to get, the packages get a lot nicer. And, by the 60s, you have transistor radios. And you guys know the rest of this story. It scales amazingly fast as things get more predictable and more precise. And so, I think when I try to think about where we are, this is 25 years ago now. And in 25 years, like I said, the circuits didn't get a lot bigger, but I think what we have is more modalities. We have proteins, it's not just transcriptional circuits, but we have many different ways, microRNAs, all these different modalities we can use and we can combine into circuits. And I think it's possible to start, I think we're at a point now where maybe the scaling can take hold and we can have reusable components and modules, as was always envisioned, but I think it's taken us a little while to get there. And if you could do that, there's really no fundamental reason that we can't make systems that accurately sense the state of the cell, that activate not only death, but maybe reprogramming or modifications of behaviors, changing ECMs and stuff in vivo. And you can even go beyond that and start to think about, what would it take to create synthetic immune systems that are orthogonal to our natural immune system, but are also based on interacting cells in the body to cure disease. I don't think these are crazy ideas because they're not, the challenges, and there are challenges, of course, but they're not fundamental. It's not that there's something that can't work inside of a cell. The challenges really are either technical, like delivery, which is a problem, or they're biological. That is, we don't fully understand all the machinery of the cell. We don't understand the full microRNA pathway yet, and we do need to interact with those pathways. But those, I think that, I guess what I want to say really, I just feel like it's an exciting time that synthetic biology is like, maybe it's some kind of inflection point right now. It's still very young, but there's the possibility of really scaling and finally capturing the sort of Moore's law kind of behaviors that we associate with electronics. Okay, so that's my hopeful hopeful thought for the moment I'm excited about. And the most important thing is let me just thank my amazing group. So this is my group last year at our retreat in a winery in Temecula. This is the gang that did these synthetic sense and response cancer circuits. Andrew really developed this, just took it from, again, like I said, almost nothing to the state it's in now. And Lucas joined, and it's just fantastic. And Shiyu, where is Shiyu? Oh, Shiyu's not on here. But anyway, Shiyu has also been an incredible presence. And our collaborators, Hao Zhu, Dan Siegwart, Stephen Moore, amazing on that project. And the gene therapy dosage compensation work was started by Mike Flynn in the lab, and Rong has just taken it to a new level, and Acacia has been amazing on the gene therapy side, and this has been a great collaboration with Viviana Guardinaru. So anyway, thanks very much.